A very good evening to all of you. My name is Prithvi Singh and I'm working as a PhD scholar at the Center for Interdisciplinary Research in Basic Sciences, Jamia Millia Islamia, New Delhi, India. The topic for my oral presentation today is Integrated Transcriptomic and Regulatory Network Analysis reveals prognostic biomarkers involved in non-small cell lung cancer and brain metastasis. Non-small cell lung cancer, abbreviated by NSCLC, has been broadly categorized into lung adenocarcinoma, abbreviated by LUAD, and lung squamous cell carcinoma, abbreviated by LUSC, with the majority of cases being subjected to LUAD in comparison to LUSC. The major risk factors involved in NSCLC progression are smoking, alcohol abuse, carcinogen exposure, environmental pollutants, and mutations. The LUAD occurs mostly in women, unlike LUSC, which is seen mostly in male population with a larger tumor size, higher mutation rate, more oncotypical mutational drivers, and has a higher chances of death as compared to LUAD. Only a small fraction of NSCLC patients are categorized at the earlier stage, which is stage 1 and stage 2, facilitating the surgical resection. However, majority of the patients are confronted with advanced stages, which is stage 3 and stage 4, where the tumor has already been metastasized and surgical section is difficult. Also, the availability of chemotherapeutic drugs is limited because the blood-brain barrier is often a cause for not reaching to the brain metastatic patients at that late stage. NSCLC accounts for almost half of the cases with brain metastasis and are associated with the poor prognosis, with a median survival of less than 2 months if left untreated. The typical gene mutation drivers, which have been classically discussed in theory for over many years, like EGFR, ROS1, ALK, and BRAF genes, are still responsible for the NSCLC metastasis to brain. But the paucity of the literature and the availability is still less, and the further information needs to be digged out on what basis the NSCLC patients at the later stage are being driven to the brain metastasis. Because even a smaller size of metastatic lesion is often associated with significant neurological damage and can often cause other linked associated disorders like Parkinson or Alzheimer's. The methodological workflow which we adopted in our study is extraction of TCGA, LUAD, and LUD cohorts, LUSC cohorts from the UCC Zener browser, followed by their pre-processing, which generally involves a batch correction, log transformation, and normalization using DSEC2 and AdJar packages for differentially expressed genes and differentially expressed microRNAs, respectively. Side by side, we also accessed GEO, which is Gene Expression Omnibus, maintained by NCBI, for acquiring the NSCLC and brain metastatic patient data, because we wanted to bridge the gap between the stage 4 and the metastatic patients, which is the brain metastatic patients. So we categorized all the LUAD and LUC sample cohorts patients into stage-wise category, in broadly into 3 categories. First is stage 4 versus stage 3, second is stage 4 versus stage 2, and third is stage 4 versus stage 1, because we wanted to know which of the genes are differentially expressed between stage 4 and the corresponding stages 3, 2, and 1. Now, the differentially expressed genes and microRNAs that has been acquired from all these 3 categories for both the cohorts were overlapped with the brain metastasis and differential and NSCLC differentially expressed genes to acquire or collect the differentially expressed metastatic genes, which we termed as DEMGs. Now, the PPI and construction of these DEMGs was followed thereafter, and the overall survival analysis, which is a vital thing in cancers, of all the differentially expressed metastatic genes and microRNAs across all the 4 stages on both the cohorts was followed. Lastly, we done the NSCLC metastatic specific 3-node FFL construction, which is actually replica of central dogma used at a bioinformatics or system biology level. And mutation analysis using cBioportal was done for the key elements in the FFL. If we look at the broader description of the differentially expressed genes and microRNAs in both the codes of LUAD and LUSC, we are talking here about the TCGA part. The broad category clearly says that in case of LUAD, S4 versus S4, S4 versus S1 category takes the majority number of deaths in LUAD, and in LUAC, S4 versus S2 takes out the majority number of deaths. Whereas in case of DEMS, S4 versus S3 takes out in LUAD, and S4 versus S1 takes case in the LUSC part. Now we overlap all these differentially expressed genes stage-wise category, which we obtained, stage 4 versus stage 1 versus 2 versus 3, with the brain metastatic dex that has been already acquired from the GEO. The upper panel shows the number of overlapping differentially expressed metastatic genes in the LUAD cohort. The bottom panel shows in the LUSC cohort. Now we took all these genes, all these differentially expressed metastatic genes, collected them as a union and inputted them to the string database to acquire a protein-protein interaction network corresponding to the highest confidence interaction score 0.9, which gives a clear picture that a total of 26 metastatic genes are involved here and linked by 26 edges. The overall survival analysis, which is shown by the KM plots here, shows that a lower expression levels of CD7, CD44, and IL6 in case of LUAD worsened the overall survival of LUAD patients, unlike ITGB1, which in case of LUSC, if has a higher expression, worsened the overall survival. This is the overall survival plots shown by the KM and KM analysis, which we done with the survival analysis using for the microRNAs, microRNAs. It shows that the lower expression levels of MEAT23B, MEAT429 worsened the overall survival in LUAD patients, whereas the higher expression levels of MEAT27A, MEAT24, and MEAT24 1 and 2, both aliases, and the lower expression level of MEAT200B worsened the overall survival of LUSC patients. The feed-forward loop, which is actually replicating the regulatory relationships between transcription factors, microRNAs, and genes, we created using the bioinformatics point of view, where we know the theory that transcription factor generally regulates the gene by acting on its promoter region, whereas the microRNAs inhibits the gene directly by repressing its activity by attaching it to a 3' UTR region, or indirectly via stimulating the TF and then activating the gene. So we acquired all the TFs from the CHIA database, and similarly, we only took here only the prognostically significant metastatic genes as well as differentially expressed microRNAs, which has been shown just in the previous slide. And founding all the links between them via database, we observed the regulatory relationship in the FFL showing 7 nodes and 11 edges. And out of these 7 nodes, 5 were transcription factors shown by the triangular red part. The magenta circular thing is the microRNA. We obtained only one prognostically significant microRNA, B23B3P. And CD44 is the differentially expressed metastatic gene, which has been linked to these 6 elements. The box and whisker plots showed that CD44 and MEAT23B3P has been non-significantly reported in case of gender as well as stage-wise category. Interestingly, both these CD44 and MEAT23B, which is the key elements in case of our FFL, are the part of stage 4 versus stage 2 category of LUAD patients. The mutation analysis, which we done by CBioportal, we choose exactly the same number of patients which we did for the differential express analysis in CBioportal. in the LUAD patients, and observed that very high amplification frequency has been observed in CD44, comparatively much lesser in case of FOS and FOSL1, and very less in TP63 patient, TP63 gene mutation. The green bar shows missense mutation is highly dominating in EOMs transcription factor. FOS and FOSL1 are comparatively similar behavior showing. And lastly, TP63. And blue is the deep deletion, which has been majorly taking part in the GLIS3 and TP63. A total of 107 patients were finalized. The result summary, which we can, if we want to make a clear conclusion here, see, the CD44 has been majorly seen as an upregulated biomarker in many tumors, and it has been reported in many literature. In our case, it showed an downregulated state. The progression from stage 4 to metastatic part can be understood in many forms. Like CD44 is a glycolytic transmembrane protein with the first 5 exons and the last 5 exons staying constant, and other exons in between due to the alternative splicing can give rise to multiple isoforms of CD44. There's an isoform of CD44 called as CD44S and CD44V, which has been downregulated and reported in many literatures, causing metastasis in breast cancer and lung cancer. One possible hypothesis that is that it causes the metastasis from the advanced stage patients is via the ERK-ZEB1 pathway. It's a mark of cancer stem cells and often plays an important role in cellular signaling. Its increased expression is usually associated with increased invasion, cancer cell proliferation, cell cycle arrest. But in our case, we observed a downregulation state. Now, the literature that has been reported earlier shows the comparison between the normal and tumor patients, in which the tumor patients shows an increased state of CD44. Now, in case we are playing the game between only the tumor part, which is we are comparing between stages 4 was a stage 1, stage 2, and stage 3. Now, the decrease in this particular CD44 expression level and going through the metastasis brain can be subjected to the regulatory load of microRNAs. Like we observed that in many literature, ME23B, ME429, in its downregulated state, which has been observed in our case also, indirectly targets the gene, which is CD44, and causes the metastasis at a higher expression level. Okay, thank you.